Hello everyone, welcome back to another class on regeneration biology. So we will learn more in detail about retina regeneration with the special reference to sonic hedgehog signaling in this class, and also involvement of some epigenetic factors such as histone deacetylases involvement in regeneration. And I will be presenting data published from my lab. So one of the articles is published where we studied the involvement of hedgehog signaling and how LET7 microRNA influence. So those who are interested can read this paper. And the injury model we have discussed, which is done mainly through a needle poke from the back of the eye, and it's a focal injury, very easy to perform, and you can analyze the neighboring tissue. So if you look into the cartoon of regeneration, you can see soon after injury, the cytokines will be released from the retina, which will be sensed by the Muller glia and causes a proliferation and restore the normalcy. And this picture also we have seen how the Muller glia reprogram into the Muller glia derived progenitor cells, and how various signaling like Wnt signaling, FGFR signaling, insulin signaling, HBEGF signaling, they stimulate the Muller glia to get into a progenitor cell. And also the delta notch signaling restrict the zone of proliferation, and various other factors such as INSM1A, which is a negative regulator in the sense it's a transcriptional repressor. It is necessary to push the progenitor from exiting the cell cycle, from push the cell into exiting the cell cycle. And Lint28 is a negative regulator of RNA, LET7 microRNA. It's an RNA binding protein. And various factors like ASCL1A contribute to the regenerative response. And LET7 microRNA is a great deterrent of this Muller glia becoming the progenitor, because my LET7 microRNA retains the integrity of a neuron, means it will keep it in a differentiated state. If you want it to move into progenitor state, LET7 should be downregulated. So this is a retinal injury and the in situ expression of Lint28. And you can also see normally in a published paper, you will see a block area. So whenever you see a square, you should understand it's a part of the retina. And this is the injury spot, which is highlighted. And you can see the Lint28 is a major blocker of the LET7 microRNA, and Lint28 is one of the pluripotency factor expressed in the embryonic stem cells and the cancer cells. And Lint28 is found both in the nucleus and cytoplasm, that is capable of regulating the LET7 microRNA levels. One option is LET7 is present in various parts of your genome. It is also embedded in the introns of other genes. So LET7 gene, when it is produced in the form of pri-LET7, primary LET7, which is acted upon by DROSA and DGCR8 to make into pre-LET7. By chopping of this region, it will become pre-LET7, and which is exported into the cytoplasm from the nucleus, and it is processed by dicer to make mature LET7. So this pri-LET7 bound DROSA and DGCR8 will be blocked by Lint28, one option. And also LET7 microRNA will be actively degraded by the Lint28. So Lint28 acts in two phases. One is prevent the formation of the pre-LET7, and also it will prevent the stability of the mature LET7 RNA. And the downregulation of LET7 is necessary for regeneration, because the previous publications have shown that various regeneration associated genes, even though they are induced, they will not get translated into proteins if LET7 microRNA is present. So you can see ASCL1A, HSPD1, Lint28, PAX6B, KLF4, c-MYC-A, c-MYC-B, OCT4. These are all genes, they are negatively regulated via LET7 microRNA. If LET7 microRNA is missing, then this mRNA can get translated in a given cell. So now if you look into hedgehog signaling, because today's class is about hedgehog signaling, we'll come back to LET7 in a short while. So hedgehog signaling can be inactive and active. Hedgehog signaling pathway, when they are inactive, when they are not present, what happens? There is a receptor called patch that will be blocking the SMO from doing any function. SMO is the major player of the hedgehog signaling. And when the patch is occupied by SHH ligand, then it is no more able to block the SMO. Hence, the SMO will be able to inhibit a negative regulator called SUFU, because of which the glee will be able to go into the nucleus and turn on various genes. But usually when SUFU is present, it will block the gli from going into the nucleus when it is inactive Hedgehog signaling. This is active Hedgehog signaling. So this is the information you should know. You should also understand there are ways in which you can prevent the migration of SMO from the cytoplasm to the nucleus. So that also will prevent the Hedgehog signaling. So understand, SMO receptor is the actual player of the Hedgehog signaling. Patched is basically a blocker of SMO from acting. So SMO, when it is free, when it is relieved from the repression of patched receptor, it will inhibit the SUFU from its ability to block gli. As a result, the gli will go to the nucleus and turn on various genes. So Hedgehog signaling is turned on in the various time points post retinal injury. If you see, almost all of the genes are induced in the retina, and the expression pattern is not important for us as of now. And if you pick up a few of the Hedgehog signaling components such as SHH-A, SMO and patch, you can see they are induced right from 1 day post-injury, 2 day it is high, 4 day it is high, by 7 day it comes down to a normal state. This indicates the QPSCR analysis also confirms the gel picture data. So now, if you block the Hedgehog signaling using a drug called cyclopamine, which is present in Californium lily, if it is eaten by pregnant goat or any animal, then they will end up getting cyclopia, because Hedgehog signaling is a developmentally essential gene. So we checked in zebrafish, if you give cyclopamine, do we get a cyclopia? So the interest of doing this experiment is to know that what concentration we can use cyclopamine for retina regeneration studies. So we found around 30 micromolar of cyclopamine. If you give, you end up getting a fused eye called cyclopia, and you end up also getting lot of cell death, or the animal survival is compromised because of the cyclopia. So now cyclopamine, when you deliver into the eye, what you are seeing is there is a drastic decline in the progenitor number, the retinal progenitor. So this is the proliferating retinal progenitor, that is Muller-Glier derived retinal progenitor. Their number is stained with a proliferation marker such as proliferating cell nuclear antigen, PCNA it is called, and their number is decreased. This is the quantification of that. So that indicates in regeneration, for the normal proliferation to occur, we need normal Hedgehog signaling. So another approach we do is, if you want to knock down a gene, you can take zebrafish anesthetisis, deliver the Morpholino into the vitreous, and then give an electric shock so that the Morpholino will get into the retina, as you can see here, and then you can do analysis. So this, once you do this electroporation and gene knockdown, you can give BRDU pulse, and then you can sectioning, and you can image for Morpholino, immunostain, etc. That is the workflow of gene knockdown studies in the zebrafish retina. So this is one image that shows if you knock down SHH using antisense Morpholino, what you find is you see a decline in the progenitor proliferation. This is control Morpholino that do not knock down any gene, and you can see normal proliferation, and this is the merge. So the lissamin tag is the tag associated with the Morpholino. So wherever there is a red color, that means it is because of the presence of the Morpholino, because it has got a fluorescent tag. So SHH-A Morpholino, you can see how much decline in the cell number is seen. And on the other hand, if you knock down SHH, but you supplement with the SHH protein, then you rescue the regeneration. So it tells the decline in the proliferation was because of absence of SHH protein. So same way, the same knockdown condition with control Morpholino or even cyclopamine treated and SHH-A Morpholino and also the gli Morpholino. You remember the gli is the actual player of SHH signaling or sonic Hedgehog signaling in the nucleus. So you can see one of the important genes, that is ASCL1A, its level is quite good in control Morpholino. With the cyclopamine, its level goes down. With SHH-A Morpholino, level goes down. With the gli Morpholino, the level goes down. So not only the proliferation is going down, but the regeneration associated genes also going down. And another double in situ we performed using the patch 1 and also SHH signaling. Remember, the patch expression is regulated via Hedgehog signaling itself. So if the Hedgehog signaling is turned on, then the patch expression is positively regulated. So it's actually more patch in the cell, then the SHH will be sequestered by these patch, overexpressed patch. So it's the way of regulating the Hedgehog signaling itself. So you can see patch expression, lin28 expression, and the BRDO expression they are in the same cell. So lin28 expression happens in the patch expressing cell, which in turn is because of the Hedgehog signaling. So we looked into the promoter of the lin28 gene for various GLI binding site, where the actual transcription factor GLI can go and bind. We saw several binding site which indeed bind with the GLI. When you performed a GLI specific antibody, we saw this is chromatin immunoprecipitation. So these sites, when it is site 1, site 2, when they are present, they have the ability to bind, especially the site 1 closer to the ATG start site. There is a strong binding of the GLI protein, that is the chromatin immunoprecipitation. What you do is you break the DNA, you fix the cells, fix the DNA and break the DNA and pull down using the GLI antibody, and then simply do a PCR. If unbound one will be washed off, the bound one will be retained, and this is a forward primer, reverse primer, and that is what is gel is been done. This is chromatin immunoprecipitation. And after proving that lin28 is bound by GLI protein and indeed regulating the Hedgehog signaling, we looked what is the expression of SHHA protein, and we saw good expression of SHH protein in the proliferating cell. And interestingly, these proliferating cells are the one which is having a down-regulated LET7. And like I told you, down-regulation of the LET7 happens because of the lin28 expression. As you can see here, these proliferating cells have got a reduced expression of LET7 microRNA. And this LET7 microRNA, what we then next did was we analyzed whether the LET7 microRNA binding site is there in the component genes of the Hedgehog signaling. We looked for SHHA, SHHB, patch, and SMO. We found several LET7 binding site in these genes, and we made a construct where the GFP is fused with this LET7 binding regions of various SHHA, B, etc., and then overexpressed in a cell line along with the LET7 microRNA. So this is the workflow. CMV promoter, GFP, and the microRNA binding sequence of various genes and transfected, and also cells will be lysed and Western blotting assay will be performed. What we found was SHHA, B, SMO, and patched, as the LET7 microRNA concentration is increasing, you see a decreased proliferation. So what you know, the activation of Hedgehog signaling must be accompanied by lin28 and the downregulation of LET7, because it is needed for their proper translation of this protein. So if the SHH signaling has to sustain, then the lin28 becomes very important, because you have to get rid of the LET7 microRNA. So in summary, we can say that the SMO migration, if you block with the cyclopamine, the Hedgehog signaling can be blocked, but the usual mechanism is SHHA, patch 1, SMO, and the GLI. And this is the actual player. The GLI can turn on lin28. It can also influence the ASCL1A production. ASCL1A can induce the lin28 production, and lin28 negatively regulates LET7, and LET7 can negatively inhibit lin28 and also ASCL1A itself. So this is basically a cycle that occurs, and the LET7 can negatively regulate SHHA, patch, and SMO also. So LET7 must go off from the progenitor if the Hedgehog signaling has to turn on. That is the story from that paper. But how SHH is being actively secreted, or who produces it at first, is a interesting question. So these are other two papers published from our lab. One is talking about how lin28 and MYC are interplay in governing the Muller-Glier derived progenitor proliferation at different stages, and another is about the how histone deacetylase comes into picture, how HER4 and lin28 axis comes into picture. So these two stories, those who are interested can read it, and I will give a gist of it. So HDAC is histone deacetylase, and histone deacetylase, the enzyme's main role is to remove the acetyl group from the histone protein. It can deacetylate various other proteins also, But when histones are acetylated, the chromatin will be loose. The gene expression can happen. When it is deacetylated, they will make a compact chromatin. The histone will bind strongly to the DNA and the chromatin will be condensed. So if acetylation happens in the histone, gene expression can happen effectively. So this is the epigenetic role played by HDACs. Different HDACs have been looked into, and our main focus was on HDAC1 because HDAC1 is a primary histone deacetylase molecule. So if you look into the expression pattern of HDAC, what we see is this is a proliferating pCNA marker and this is the BRDU marker, and you can see the expression of HDAC is neighboring near to the proliferating cell. And the immunostain of the HDAC, you can see the BRDU positive cells do not have HDAC expression, whereas the other cells do have. So what it tells you that HDAC is secluded from the actively proliferating cell. That means you don't want deacetylation actively happening in the proliferating cell. That is why HDAC is missing. If HDAC is present, it will deacetylate it and chromatin will be compact. So from this observation, this is what you can get it. However, there is a catch. If you look HDAC, which is excluded from the proliferating cell, but if it is associated with another protein called MICB, MICB can influence the lin-28 expression in both ways. Either it can be an activator or it can be a repressor. Let us see how. MICB normally binds to the EBOX binding region, or MICB. Normally around 25% of the total polymerase 2 transcribed genes have got MICB binding site. So MICB activating lin-28 is just normal thing, and in the paper we have also shown, confirmed it by chromatin immunoprecipitation also. So the MICB is present in the lin-28 expressing cell, as you can see here. But interesting thing, one thing you should see is in one cell where is MICB expression is there, lin-28 is missing, and but it is a proliferating cell. You can see BRDU is there. So what made this cell not to have lin-28? So which made us to believe that there is a possibility that MICB, although present, is not activating the lin-28. So we later found in this study that the MICB can interact with HDAC and bind on to the lin-28 promoter and cause a repression of gene expression. So HDAC1 can normally be seen missing from the proliferating cell. So here proliferation is there in both the cells, but MICB is present but lin-28 is missing. So this is because MICB can choose a partner, whether it should be HDAC or which can cause a repression on the lin-28 promoter, or it can act as an activator. So HDAC knockdown blocks the retinal progenitor proliferation. You have used two different Morpholino, two different HDAC1 Morpholino and second target, two targets we used. And in a concentration dependent manner, you see a decline in the pCNA expression. Like control Morpholino, there is a normal proliferation. HDAC, both the concentration, HDAC2, both the concentration, you see a decrease in the proliferation, as you can see here. So how this action happens is histone acetyl transferases add acetyl group onto the histone protein, whereas HDAC does the reverse. So if you do HDAC inhibition, various chemicals are there, trichostatin is there, valproic acid is there, you can block HDAC, and we explored what happens if you block HDAC to the regeneration. So we found that HDAC mediated pathway, when you block HDAC, we see a decrease in the lin-28 expression. As you can see here in the qPCR, there is a concentration dependent decline in the both with the TSA and valproic acid. They are two inhibitors of HDAC. We see a decrease in the lin-28 expression. And you can also see the same decline in the RNA in situ hybridization. Lin-28 expression is decreased because of the HDAC inhibition. Accordingly, proliferation also is decreased. And there is, since the lin-28 level is decreased, there will be an increase in the LET7 expression also. So the LET7 levels do go up. And these LET7, you should understand, they don't allow the translation of various regeneration associated genes. They don't allow the Hedgehog signaling components also. High LET7 means whatever it is, the protein translation will not happen. So the increased LET7 could account for why there is a decrease in the proliferation because of HDAC inhibition. So HDAC inhibition mediated reduction in proliferation is reversible. Like in various regeneration studies, we have seen that in fin regeneration also we have seen it, like FGF20 A, if you block it, the wound heals, but there is no blastema formation or regeneration. We have seen follistatin knockdown in planaria. If you knock it down, you get a wound healing, but the planaria will not regenerate. We have seen that. But interesting thing here is, in retina, what we looked, if you remove the blocker, so when you have a blocker, trichostatin or valproic acid, when it is there, you don't get regeneration. So what we did was we blocked up to 4 days. 4 days is the peak of proliferation in an injury method. So we removed the VPA and until 4 days, no regeneration. As you can see here, if you put it, then you don't find any proliferative response. Then what we did was we removed the VPA and waited up to 8 days, means another 4 more days. Injury was given in 0 day and put in drug for 4 days, and 4th day we put the fish in normal water, and then you allowed it to grow up to 8 days. What we found was the regeneration bounces back. So there is an increase in the PCNA proliferation, and this is simply because the retina is so plastic that it knows how to come back and how to bounce back and how to restore its damaged portion. So that was something very interesting, that unlike planaria or unlike zebrafish fin, if you block HDACs, there is a chance that the animal will bounce back. And another interesting thing, during this re-entry, we also looked for Lin28 expression. What we found, that once you withdraw the drug, put 4 days in the drug and then stop it. That means don't inhibit the HDACs. When you withdraw it, you will see an increase in the expression of Lin28. And naturally you can expect the LET7 levels will go down, which also we have checked it and it is there in the paper. So the LET7 levels when goes down, the regeneration associated genes can happily get translated. So this is the increased level of Lin28 A, and this is the qPCR of Lin28 level goes up. So what you can understand from here is that there is an increase in the how Lin28 and LET7 is influencing and regulating. In this picture, what you can see here is SHH follow this workflow. We have seen this SHH-A, patch 1, SMO and GLI. So patch 1 is a negative regulator of SMO and SMO is a positive regulator of GLI, because SMO's role is to get rid of the repression of GLI by SUFU. So the moment SMO is free, it will recruit the SUFU and free the GLI. That is what it goes to the nucleus, and GLI helps in the activation of Lin28 and also various other genes, not just Lin28. We are discussing only about Lin28 because its implication in the LET7. And it can also activate ASCL1A. And one of our earlier stories, we have shown ASCL1A can activate Lin28. And Lin28 is the major negative regulator of LET7. And LET7 when present in a tissue, it simply will not allow the translation of various genes. You have seen it, various genes of which is kind of associated with the regeneration, and especially the hedgehog signaling. And the same LET7, I will show that picture once again quickly so that you will remember. Same LET7 can negatively regulate Lin28 and it can negatively regulate ASCL1A. And here what we show was the HDAC can combine with the MIGB and it can negatively regulate the Lin28. MIGB alone positively regulate Lin28. That is one way. Apart from ASCL1's role, MIGB also can positively regulate Lin28, but it can influence in the HDAC expressing cell. HDAC expressing cell, even if the MIGB is expressed, it will not allow an activation of Lin28. If Lin28 is not activated, what will happen? The LET7 level will stay high, and because of this, there will not be any translation. I will quickly show that LET7 regulation. Here, So this is a earlier paper which I published where ASCL1 is negatively regulated by LET7 and also LIN28 also negatively regulated by LET7. So even though these are all regeneration associated genes, they are essential. These are all pluripotency factors, and KLF and OCT4 is little marginally down regulated compared to the control. But whereas these genes ASCL1, HSPD, LIN28, etc., they have binding site. Various regeneration associated genes may not have binding site, but they are not good enough. Say there are 5 genes or 10 genes required for regeneration, but 6 of them have got LET7 binding site. Remaining 4 is not good enough. Even though they are translated, they are not good enough or sufficient for causing a regenerative response. So this is the idea you should keep in your mind. That is why in the last model, we have shown that ASCL1A and LIN28, they negatively regulate each other. And the c-MYC itself, that's another thing we should keep in mind. The c-MYC itself is negatively regulated by LET7 microRNA. Now, if you go into, and another thing we have seen is this LET7 itself regulating the, so this one is the LET7 which is going off from the proliferating cell, and credit goes to the LIN28 expression. You can see this, the neighboring cells have it, they don't proliferate. As you can see here, this is the merge. So this is the non-proliferating cell, so they go off. And whereas in uninjured retina, you see the entire inner nuclear layer and the ganglion cell layer has got the LET7 expression. This is the RNA in situ hybridization. Then comes this how hedgehog signaling components SHH, A, B, SMO and patched are regulated. Here, this is basically MRE mutant, where the microRNA recognition element is mutated in this construct, where you have got the normal protein, and whereas in these ones, the protein levels are down regulated. That is why it becomes so foolproof that its levels are influenced exclusively by, or the protein levels are influenced exclusively by the abundance of LET7 microRNA. So based on that, we have come up with this model, and so that is why we can see here the HDAC expression is there everywhere but in the proliferating cell. Because if HDAC expression is there, even if MYC is present, it will not cause a proliferation because the LIN28 expression is negatively regulated. And in this model, you can see that is what we have discussed here, that the MYC-B is negatively regulating the LIN28 expression. Then another story, I will not go into the details, those who are interested can read, is we found another transcript, another tumor suppressor called PTEN that associates with various gene regulatory events that is essential for retina regeneration. So soon after injury, just like HDAC is going off, the PTEN also goes off from the proliferating progenitors, and this is necessary for activating the AKT. So PTEN, when it is super repressed, like you got rid of PTEN from various other cells which normally don't have to get rid of because it is not a responsive cell, then you find too much of a proliferative response. So those who are interested can read this article that is published in the GLIA. And also we also looked for the role of TGF-beta signaling. In mammals, the TGF-beta signaling is pro-proliferative, anti-proliferative, whereas in zebrafish, TGF-beta signaling is pro-proliferative. So this is something very interesting. Those who are interested can read this article also, which is cited here. And we also looked for how OCT4 is influencing, which is a pluripotency factor, and how it is influencing the regenerative response at different phases, like influencing the epithelial mesenchymal transition or mesenchymal to epithelial transition. We also looking for, these articles will be published soon, that is the involvement of yin-yang protein and also polycomb repressor protein CTCF, and also long non-coding RNA such as MALAT1, how it is influencing regeneration, and how NURD complex is influencing the regeneration, and also how type 2 diabetes is influencing regeneration, and also hyposignaling and neuroglin KDM pathways influencing the regeneration. So these are all the angles in which regeneration can be studied. We will study more about regeneration in coming classes. Thank you.